Hello friends, how are you all? I hope you all are doing well and I found you all in the good health. Myself, Megha, I'm research scholar at NIPGR. Here I am with a new topic that is the RNA interference. So let's get started with the new topic. So firstly, the introduction. RNA interference is a natural mechanism that is found in the most eukaryotic organisms that inhibit the gene expression at the stage of translation or it hinders the transcription of a specific gene. RNA interference is being developed and exploited to improve plants by modifying their endogenous gene expression as well as to target the pest and pathogen genes both within the plants. So, as I have mentioned earlier, also that it is a natural mechanism and it is widely used for exploited to make a major contribution toward the integrated pest management because the wider level in agriculture industries is using the pesticide and fertilizers which are very harmful for our climate and also it can lead to many diseases. For to sustainable agriculture strategies, these RNAi mechanism is a natural mechanism. It can be exploited and can target the particular genes within the plant to resist the pathogen and pest infections. So it can be a great strategy for sustainable agriculture strategies and it needed to be implied worldwide to secure our current and the future food production. That's why we need to get detail of this particular technique as it is very important in our food industry as well and many scientists are using it for studies the gene expression of particular genes and also it is widely being used in the agriculture nowadays. So history behind it. Napoleon Jorgensen were first to report an RNAi type phenomenon in 1990. They were working on the Petunia hybrida plant, which was engineered to harbor the additional transgene copies of the flower pigmentation gene that is the chalcone synthase. While introducing the additional transgene of the chalcone synthase, they notices the homology-dependent gene silencing mechanism in plant. So this homologic gene silencing mechanism in plant is due to the RNA interference. Nowadays we know that, but they were not aware of this particular phenomenon. So they just instituted is that the homology dependent gene silencing mechanism in plant. Then the further research is carried out by Andrew Fire and Alexi Mallow while they're working on the C. elegans. And for discovering the RNA interference mechanism, they got the Nobel Prize in Physiology and Medicine for their work in the RNA. So while working in the working with the C. elegans, they notices that when they introduce this single-stranded RNA, whether the sense and the antisense, there is no effect. But when they introduces the double-stranded RNA, they notice the twitching of the muscle protein, which is as same as to the worms carrying the defective gene for the muscle protein. So it is evident that somehow the double-stranded DNA is carrying out some because of some mechanism is causing the defectiveness of that particular muscle protein. And then they came to know about the RNAi mechanism. So to understand the RNA interference mechanism, we have to go on through the central dogma. In central dogma, we know that DNA is converted into the RNA by transcription. mRNA is further translated to the protein. So these protein, the further processing of mRNA to protein is carried out. So in RNAi mechanism, RNAi interference is an important biological mechanism in the regulation of the gene expression. So as we know that the non-coding genes were previously called the junk DNA, but nowadays we can't use the junk DNA term for the non-coding genes because they are also having the important role in the regulation of the gene, and it is evident from one of the these RNA mechanism because it has very important role in biological mechanism in the regulation of the gene expression. So the junk DNA. The so-called junk DNA is no more junk DNA because the non-coding genes are somehow playing the role of regulation with the help of the RNA mechanism to for the regulation of the gene expressions. So double-stranded RNA binds to the protein dicer. So first of all, this double-stranded RNA, the dicer protein is very specific to the double-stranded RNA. It binds to the double-stranded RNA and cleaves the double-stranded RNA into the smaller fragments. And one of the strand from the smaller fragments, that is called the microRNAs. The one of the strand from the mature microRNAs is loaded on the RISC complex. These strand of the microRNA guides the RISC complex towards the mRNA. It forms the complex with the mRNA by the base pairing to the complementary sequences. And it forms the base pairing, the RISC complex get activated and the argonaut cleaves the mRNA in the between of siRNA, where the in central region of the siRNA. So why there is a need for the interference? The need for the interference is a defense mechanism and the genome-wide regulation, as I have mentioned earlier. So, in the plants have the defense mechanism against viral infection, which this is the RNA mechanism, which is the defense mechanism against viral infection. It is also acting as a defense mechanism against the transpose and other insertional sequences, which hinders the expression of the coding genes. So it is a defense mechanism regarding these, and also it is playing a wide role in the regulation because it reduces the accumulation of RNA molecule with sequence similarity to introduced nucleic acid. So we concluded that it is playing a various role in the cell. It can be used by the scientist. They can make a tailor-made double-stranded RNA to activate the RNAi mechanism to target the specific genes and lower their expression, and to study them also to target the specific gene, to study their regulation, also to use them in particular defense mechanism by the plants. It is a defense mechanism against viral RNA because the viral RNA, double-stranded viral RNA, when introduced into the plant, by it is processed by dicer, then the RISC complex and protein synthesized is suppressed. It is widely used mechanism by the microRNAs in regulation because these microRNA are hairpin-like structure, which are somehow similar to the double-stranded RNA. So they are these microRNA are also processed by the dicer and the RISC complex to suppress the protein synthesis and thereby regulation of the genome and reduce the accumulation of RNA molecule with sequence similarity to the introduced nucleic acid. So, now the RNA mechanism in detail. The double-stranded RNA or RNA having strong secondary structure like hairpin like a structure or the microRNA are believed to be the initiator of the RNAi interference mechanism. Then the protein participating in RNAi are believed to be conserved across the different organisms. That means this mechanism is conserved among different organisms also. In the basic RNA silencing pathway, the double-stranded RNA or the hairpin RNA is processed by the dicer or DCL protein into the 20 to 24 nucleotide small RNA duplexes with two nucleotide at 3 prime overhang at both ends. One strand of the sRNA duplex is incorporated into the argonaut, forming an RNA inducing silencing complex. This sRNA molecule guides the risk to the complementary region of the single-stranded RNA, and the argonaut protein then cleaves the mRNA at the nucleotide corresponding to the central region of the siRNA, and RNAi leads to the degradation of the targeted mRNA and inhibition of its translation or maybe methylation of the encoded gene or a combination of the above. So next we need to have a detail about dicer. Dicer is an endoribonuclease and or helicase with RNase motif. Is an ATP-dependent ribonuclease enzyme. Being the part of the RNase third family, dicer cleaves the double-stranded RNA and pre-microRNA into the short double-stranded RNA fragment called small interfering RNA and the microRNA respectively. Typed has four distinct domains, that is a helicase domain, Pas domain, RNA is third, and the double-stranded RNA binding domain. This helicase help in the unwinding of the double-stranded RNA. Pas domain acts with the 3' overhang of the double-stranded RNA, and the RNA is third cut the processed the double-stranded RNA into the mature small interfering RNA. Then the risk complex. The RNA-induced silencing complex or the risk complex is a multi-protein complex, specifically a ribonucleoprotein, which function in gene silencing via a variety of pathways at the transcription and the translation level. The cytoplasmic RNA-induced silencing complex contain double-stranded RNA binding protein, including the organoid protein, the dicer protein, and the TRBP transactivation response RNA binding protein. Types of RNAi pathways. According to the source of the double-stranded RNA or the hairpin RNA precursor and the functional target of the sRNA, RNA silencing in plants can be classified into the four overlapping but functionally distinct pathways. First one is the microRNA pathway, second is the trans-acting small interfering RNA pathway, RNA-directed DNA methylation pathway, and the last one is the exogenic RNA silencing pathway. These four pathways are associated with the diversification of the RNA silencing pathway. Because of these diversified pathways, plants have also evolved multiple RNA silencing factors. For instance, the model plant Arabidopsis encodes four different dicer proteins and six different RNA dependent RNA polymerases and 10 argonaute protein. That evident that these silencing pathways are very diversified and plus several other factors are there. So let's get detail into these pathway. These are the post translation gene silencing pathway and the translation gene silencing pathways. So first one is the microRNA induced gene silencing pathway, where this microRNA is processed by DCL1, dicer1, and then the argonaute protein with the help of the RISC complex cleaves the RNA and leads to its degradation. Then the trans-acting gene silencing pathway, where the single-stranded RNA is converted into the double-stranded RNA by the RNA-dependent RNA polymerase, and then it is processed by DCL4, dicer4, and then further the RISC complex cleaves the mRNA at the particular site. Then the viral induced or exogenic RNA induced gene silencing pathway, where the double stranded RNA of the virus can be acted as activation of the dicer, then it further processed by argonaut and RISC complex to cleave the particular mRNA cleavage. Then the translation gene silencing pathway, where the heterochromatin or repeat regions can be uh, converted into double-stranded RNA by the RNA-dependent RNA polymerases, which then further processed by the dicer 3 and then the RISC complex for DNA methylation. It can also lead to the DNA methylation as well. So, as I have mentioned, that the RNA-directed DNA methylation pathway. So, this is the RNA-directed DNA methylation pathway. So, these are the mechanism and the pathways. Then application. RNA silencing technology are particular the HpRNA transgene technology, the hairpin RNA transgene technology, and more recently the miRNA technology have been widely used to study the gene functions to generate the pathogen and pest resistance. It can also improve the agronomical trait by manipulating the expression of the metabolic pathway genes in a variety of model and crop plant species. Limitations. The post-transcriptional RNA silencing usually does not result in complete gene silencing. So this is the one of the limitation in the RNAi mechanism to be widely used as researches and to improve the crop plants. In addition, it remains unclear if RNA silencing technology can be used to consistently switch off endogenous gene promoters in plant, which is unlike the transgene promoters appear to be resistant to siRNA directed transcriptional silencing. And as we have discussed many pathways, so the combination of RNA silencing technology based on the different RNA silencing pathway could enhance the efficiency of silencing. For instance, combining the post-translation gene silencing with the translation gene silencing based technology can lead to more efficient way of producing the improved crop plants with high defense mechanism against the pest and pathogen. These are the references which I have used. Thank you. I hope you enjoyed it, and please like, share, and comment if you like it. Thank you. Have a good day.